second membrane started? In step 4?
Yeah.
The fourth picture? What does it say at the fourth picture?
Cytoplasmic membrane grows
It grows.
and engulfs spore within a second membrane.
Yes.
The cell's DNA disintegrates.
Okay, so the second membrane is now coming about. The DNA, the original copy of DNA is being disintegrated. And so now it's set up to do what? To start forming that spore coat.
So that will be step 5?
Step 5 coming up, yeah.
Is when it forms.
Okay, in step 5, what does it say?
The cortex of peptidoglycan is deposited between the membranes. Meanwhile, the quality
Diploconic?
acid and calcium ions accumulate within the center of the endospore.
Okay, so. Now you have
Can I just draw forever?
this is not an art class, we'll just draw things.
So the first step has the division of cells A and B, and then the second step is DNA replicated and will line along the cell's axis.
Okay, so
I just put DNA.
in that step, you're going to have calcium ions and diploconic acid that will come inside this inner area, right here, right? In between these two membranes, you're going to have peptidoglycan going into there, right?
Okay.
So you got peptidoglycan in between those two membranes, right? Kind of like forming a cell wall, right? And then inside here you have diploconic acid and you have calcium ions. That's kind of like your preservative that's in there. So now you have a preservative for your DNA, and you have a peptidoglycan coat on the outside which is protected.
Wait, can you say the middle part again? Like you said, peptidoglycan in between the.
Okay, peptidoglycan is between the first and second membrane,
But
right?
after that, the middle.
The diploconic acid and the calcium ions are now going to go inside that inner area. And they act like a preservative inside there. So, at this point, that spore becomes mature. The spore coat matures, right? Once that spore coat matures, what characteristics does it take on? Why do we have to boil spore-forming bacteria for more than 20 minutes at a high heat?
They're resistant.
Because they're resistant to high heat, okay? They're also resistant to chemical insult or chemicals coming in. These chemicals cannot get inside there. Why do we have to take our spore-forming bacteria and apply Malachi Green over boiling hot water? That boiling hot water allows for that stain to get inside here,
You put
right?
vegetarian growth and
So,
Put these two
therefore,
together.
it's normally resistant to high heat. So I have to use very high heat and that steam to get that dye inside there. So knowing that from your lab, you should know the characteristics of a mature spore. It's resistant to heat and it's resistant to chemicals getting inside there. Once that happens, then the mother cell will disintegrate and leaving this finished spore out in the outside. That's why when we do the spore stain, we see pink cells or pink debris. Sometimes you look at all this pink debris that's out there, but you see the little spores, like the emerald spores that are there, sitting on the outside, because that cell has now been degraded. But the cell's still gonna be, the cell debris is still gonna color pink, but the spore is gonna be colored green, because we drove that dye inside there with the heat and the steam. Okay? Anybody confused on that?
Mm-hmm.
What are you confused about?
Never mind. The whole thing. I'm just gonna have to memorize it, the steps.
Okay.
I had just put,
It follows along the book except for the first part.
I had started off putting endospores form when nutrients are low, they can survive extreme conditions, such as your high heat.
Okay.
Endospore starts forming and the DNA replicates. It then forms a forespore and the second cytoplasmic membrane surrounds it. And then that's all I had got to. Then I'm gonna have to break down the rest of the steps
Okay.
into simple as.
Shouldn't be that bad.
Okay. Now, the next question deals with, of course, eukaryotic cells, right? Eukaryotic cells such as your immune cells that are inside you. That means we're looking at endocytosis. There's 3 types of endocytosis that we talked about, right? First one we talked about was pinocytosis or penocytosis, however you want to pronounce it. Second one was receptor-mediated endocytosis. Receptor-mediated. Receptor-mediated. The receptors mediate the entrance of the ligand into the cell. That's like you go into Walmart, and there's a greeter there, and if it's a good greeter, he greets you, asks you, what do you want? You say, I want to find yogurt. So he will grab you by your shirt and drag you over to the section that is where the yogurt is at, right? Receptor mediated, okay? The other one is phagocytosis. Morning. Did you think it was 11 o'clock too?
No, I thought it was 11 o'clock, like I didn't know that it was 10, I thought it was 11. I'm just.
Okay, so pinocytosis, receptor-mediated endocytosis, and phagocytosis. Now, the characteristics of pinocytosis is basically the cell invaginates and forms a endosome. Inside that endosome is a liquid sampling of its outside, right? This is something that occurs like in your digestive system, where you get pinocytosis inside your intestines, right? So it takes a liquid sample of what's out there. That endosome, which is a vesicle, is now inside that cell, right? So the interior of the cell. You have another vessel that's called the lysosome. Lysosome contains enzymes. And we know what enzymes do, right? What do enzymes do?
Break stuff down.
So these break things down. So you get a fusion of that endosome with the lysosome. The contents of that lysosome will enter inside there and start breaking down whatever stuff is in there, right? So kind of like digesting in your stomach, basically. The lysosome is the stomach of the cell. So it breaks that stuff down. Once it breaks it down, any waste that is comes from that needs to get outside the cell, right? And so that endolysosome fusion right there, that one vesicle now will move to the other side of the cell and then be released through exocytosis. Okay, so you have endocytosis, the entrance, you have the processing of it on the inside, and you have exocytosis. This whole process is called transcytosis, meaning transitioning through the cell. Transcytosis is the whole process. So that is pinocytosis. So the basic understanding here is you have 3 different types, right? Once that vesicle enters into the cell, the same thing is going to happen to it no matter what. Okay? The only difference is how it gets in. How it gets in with pinocytosis, you have invagination of the water or liquid portion out there, brings it in. Receptor-mediated endocytosis, you have specific binding of a ligand, L-I-G-A-N, specific binding of a ligand to a receptor. Once that binding happens, that causes the cell membrane to invaginate, just like it did with pinocytosis. When it invaginates, it's going to form an endosome, right? That
What did you say at the beginning?
receptor-mediated, specific binding of a ligand.
Of what?
Ligand, L-I-G-A-N. Okay, specifically binds to it. That causes the cell membrane to invaginate. Once it invaginates, it's going to form an endosome, just like it did with pinocytosis. What's going to happen to that endosome next? Same thing that happened to the endosome in pinocytosis, right? You have a lysosome that binds onto it, fuses it. The enzymes from the lysosome digest that content that's in there, and the waste gets put out through exocytosis. Right? Why are you looking at me like that? You have a question on it?
No, I didn't get it.
You got it? You didn't get it? Okay. What part did you not get?
No, it's because I kind of came through the middle. No, it's because I came through the middle of it, so I didn't get it.
Bless you.
Okay. We're talking about how things get into the cell, endocytosis, right? There's 3 types. Pinocytosis, taking a water sample.
Okay,
Coffee.
the essay question reads, describe the process of endocytosis with examples of authored types.
Yes.
On our essay question, if we describe the endocytosis occurring like when the cytoplasmic membrane surrounds a substance and brings it into the cell, right? And then we can describe the phagocytosis, like bringing a solid into the cell, pinocytosis, bringing a liquid into the cell, exocytosis, the reverse of endocytosis, and it exports substance from the cell. So does that cover all of it? Good enough.
Okay, so let's see.
Or, and then we have to put the receptor mediated.
Endocytosis is the act of a cell bringing act. It is active, it is live, it is doing something. Act of a cell bringing a substance